Good morning again. Thank you for joining us at our annual Benefactor Campus Experience event and today's classroom experience entitled Wearables and the Future of Healthcare by our dear Professor Teck, who is with us today. My name is Andrew from the Development Office, and I will be your MC for today. On behalf of the NUS Development Office, I would like to extend a very warm welcome to all our guests here and our benefactors. It's really good to see everyone again, and we appreciate you taking the time to spend your Saturday morning with us, despite the inconvenience of this ongoing pandemic. So thank you very much. The Benefactor Campus Experience event is an exclusive event that we designed for you, our benefactor. We have continuously and generously supported us over the years, and because of the support, that the university is able to thrive in its mission of achieving greater excellence. As much as we would have liked for you to experience the various impacts of your support that you have brought to the university, but unfortunately due to COVID-19 situation and for your safety, we have to scale down the events for this year. Now I would like to introduce the speaker for our today's class. So Prof. Lim is the inaugural NUSS Chair Professor at the Department of Biomedical Engineering and founding Principal Investigator at the Mechanobiology Institute. He is also the Director of the Institute for Health Innovation and Technology, iHealth-Teck. Prof. Lim co-founded the Department of Biomedical Engineering, the NUS Nanoscience and Nanotechnology Initiative, and the Center for Advanced 2D Materials. Prof. Lim himself has authored over 400 peer-reviewed journal papers and delivered more than 390 talks. He is also an elected fellow of the American Institute for Medical and Biological Engineering, the International Academy of Medical and Biological Engineering, and the Academy of Engineering Singapore. Prof. Lim himself has also co-founded one incubator and 6 startups which are commercializing technologies developed in his laboratory. So with that, it's my pleasure to welcome Professor Lim. Prof, please.
Okay, good morning, everyone. Firstly, thank you, Andrew, for your kind introduction. So when the organizers actually approached me to give this talk, I said yes immediately because this is actually my first face-to-face lecture in front of a live audience instead of speaking in front of a screen. So I really appreciate and really treasure this opportunity. The other thing is also, I think this is also an opportunity for me to share with you also what we are doing in our lab. And in particular, I think the donations you have given actually did fund some of my research, in particular the COVID research I'm going to talk afterwards as well. Before I go into the actual lecture itself, I think normally sometimes when we organize events like this, we do have donors like yourself come physically to our lab or institute. Unfortunately, because of this COVID-19, we can't do that. But as Andrew has mentioned, I'm currently leading an institute called the Institute for Health Innovation and Technology, or in short, iHealth-Teck, and it's actually a university-level research institute. And what I'm going to do is to briefly tell you a bit about this institute. We started about 3 years ago, and really the whole idea or the vision of the institute is to advance health through innovation. And we have four mission statements. One is to identify what are the current clinical unmet needs and challenges. And then second is to see if we can innovate and come up with some very disruptive and very useful health solutions and technologies. But importantly also, we want to incubate these technologies because we want to bring them out from the bench to the bedside and hospital. So we do try to not only incubate, but work also with the commercial partners to bring them out. But importantly, we also want to bring impact to patients, doctors, and also society. So in terms of impact, what I'm talking about is to improve not only health span, but also longevity. You know, Singapore actually is ranked one of the highest in terms of life expectancy. But in terms of health span, many of us will spend average about 10 years in poor health. And so we want to actually shorten that. Of course, we want to also come up with better disease diagnosis and monitoring devices and technology as well, so that we can detect the disease early. And as a university, we also want to train our students to become future leaders of this healthcare workforce. And in fact, this COVID-19 shows us that this is actually a very important area for us to focus on. So in our institute, what we aim to do is three things. One is prevention. If we can prevent the disease from occurring, that would be the best. Then we don't need any treatment or whatever. But if someone do contract disease, then we try to detect them at a very early stage. If we can detect them early, then we can better treat the patient. But if we don't, and the person is sick, then we will try to go for what we call precision therapy, because all of us are very different individuals. And in one of the areas I work on in cancer, we found that no two patients are the same. And one of the reasons is because of the makeup of our body. In fact, the composition of the cancer cells varies from person to person. And that means that we have to tailor the treatment according to the individual. So this is just a snapshot of all the different technologies we have developed in our lab. But in short, we have three focus areas. One is precision medicine. Second is smart sensors and AI. And then the third is mental health and aging. So if you look at this whole different technologies, we not only develop wearable devices, but also other different technologies. For example, e-skin prosthesis enable you to sense or to have the sense of touch for people who lose that. To developing insole sensors for diabetic foot ulcer management. So even creating, for example, microfluidic devices to grow patient-derived cancer cells so we can do drug tests to see what are the appropriate treatment to administer to the patient. So I won't be able to go through all of them, but hopefully through this talk, I will cover some of this technology. And right now, we are located in the School of Medicine. So we are given one floor at MD6, but well, actually, it's supposed to be ready by this quarter, but because of COVID-19, it's going to be only TOP in early Q1 of 2021. But hopefully, by the time we can move into a new building called the Engineering in Medicine building, and this building is actually located in Faculty of Engineering, and it's going to be an 8-story building where we have a concentration of researchers working in the area of medical technologies or health technologies. And so when it's up, I certainly welcome you all to come and visit us. Maybe you can hold a visit to our institute and show you some of the research we're working over there. So for this talk, first, because we're talking about wearables, I will first give an introduction about what wearable is all about. And then I will showcase some of the wearable sensing technologies that we developed in my lab. And then I will just briefly comment about the sensors of the future. And then I think the organizers wanted me to also cover some of the COVID-19 test kit that we've been developing in my lab. In fact, we've been very busy since circuit breaker period. In fact, myself and my team has been coming back every day since circuit breaker started to try to come up with these two test kits. I'll show you this, what we call point of care test kits. That means they are devices or test kit that can test patients or individuals right at the point or on site where they are, rather than to have the sample bring to a centralized lab to test. Okay, so as we all know, many of us are actually already wearing wearables. So I myself, actually, I'm wearing an Apple Watch, and I use it quite a bit for exercise and so on. But we are really entering into the age of wearables. In fact, we are only just at the dawn of this age of wearables, and there are many exciting things to come. And in fact, if you look at the wearables itself, it can cover many different areas, from measuring our vital signs like heart rate, blood pressure, to even being able to measure your gait, as you run, how much pressure is acting on your feet, to even hearing aid as well. So you also have devices where you can assist you in hearing. And so we are really entering into the age of wearables. And in fact, prior to the age of wearables, right, I mean, many of us are always, one of the things that we are very concerned with is our health. And most of the time, we'll go for health screening. And it's always done maybe once a year. you get a time point. Let's say every January you go for a health screening. You know what your health status is. Then the next year you come back again. You go for health screening, and then you'll see whether certain biomarkers has been going up or down with regard to your health status. But with the wearables itself, the idea is to be able to monitor your health or your well-being on a continuous basis. And this is actually what many researchers or companies are trying to do. And that's one reason why we are wearing fitness tracker and so on, to track number of steps we take and at the same time to provide nudges to us to adopt a more healthier lifestyle. But moving forward, later on when I talk about sensors of the future, it's going to be quite different, because right now we are still talking about wearables, a device where you have to wear, be it on the wrist or embedded into your jacket or your shirt. And this is a very busy chart, but if you look at this chart itself, there are many devices now being developed around the world. And this chart is to show you that the areas that they cover not only includes safety and security, but also medical, wellness, sports and fitness, lifestyle computing, communication, and even glamour. That means fashion and so on. It's a very hot area now. A lot of people are developing many wearable devices for many purposes. And so, if you look at this chart, it's just a lot of effort has been put in to develop many devices, but obviously not all will eventually be adopted for use. And this is obtained for Teck's research, and it shows how we are moving. We have a lot of consumer tech, but at the same time, medical is also another very important area to go into. So this is actually up to only 2019, but already you can see the potential of wearable devices, not only for non-medical, but also medical use. And it will always be on the upward trend for the next 5 to 10 years. And just to show you what some of the ongoing research that's happening. So, for example, there are some interesting wearable devices like embedded in clothing, and there's actually one interesting research I was reading about breast cancer detection. So there are researchers actually coming up with a bra-like apparel where for example, women can wear it and be able to monitor if there's actually a tumor inside the breast. And of course, wristband is nothing new. All of us already, some of us are already wearing it, but to be able to measure glucose will be something very new, because right now glucose, you have to do a finger prick, or there are devices where you can attach to your skin, but if you can do it to a wristband watch, without this invasive means of detecting, for example, for diabetic patients, that would be very good. And shoes is something I'll show you afterwards, because we are also working on technology like this. And contact lens is also something that is also pretty hot now. Many people are trying to develop contact lens that again can monitor, for diabetic patients, glucose level and so on. And breath analysis is, all of us are wearing masks, right? And there are researchers that are trying to develop masks that can detect COVID-19. But at the same time, people are also developing masks for other purposes. To analyze your breath. In particular, I have a colleague who is looking at developing a breath analyzer to detect lung cancer, for example. In fact, one of my ex-students who I'm supervising, she has started a company called Breathtonics, and she is actually trying to use this device to detect COVID-19 from breath of patients. And of course, electronic skin is also another area that some of my colleagues are working on. Not only to detect vital signs, but also to be able to give the patient a sense of touch if they lose that. And then implantables are more invasive, but something that is implanted into your body to measure cardiac signals and so on. So moving forward, I will actually talk about some of the wearable sensing technology that we've developed, and for this case, here in NUS in my lab. So when a few years ago, I have a clinician who come to me for NUH, and he was telling me that in the rehab lab, they are using body sensors that are very rigid, very bulky, and very fragile. And he wanted me to see if we can come up with something that is very lightweight, very simple to use, that can be attached to your body. And so we were thinking about whether we can come up with something that's easy to use. And so So we first look at what is existing out there in the market. And if you look at wearables, right, there are actually three components. One is the sensor itself. What is the sensing element that you're using to detect signals from the human body? The second is the electronics and circuitry. Because once you are able to sense the signal or the data, then you want to be able to capture it and then be able to send it either wirelessly or wired through to your computer or wirelessly to your cell phone or tablet. Then second, the third one is actually the substrate itself. Because you are going to mount it on your skin, so it has to be a material that's not stiff and rigid, that has to have the same property, hopefully the same properties as that of your skin. So you can comfortably mount it on your body, without even feeling it if it's very small. So in my lab, I think some of you have also attended my previous talk. One of my expertise is in microfluidics, and previously we have been developing microfluidics for detection of cancer cells, for example, and we actually commercialized this. The company has already gone public 2 years ago. So because we have microfluidics, so we thought, can we develop something that is using microfluidics as a wearable device? And which means I will have to do three things. One is the sensing element will be a liquid rather than a rigid solid sensor. And most of the time, a lot of publications, if you look at, they all use solid sensors. So for us, we use liquid as the sensing element. Then second is the material itself. As I mentioned, it should have skin-like properties so they can be easily mounted on your skin. And you can feel comfortable. And third is the electronics, where we will have to capture the data and then transmit it wirelessly. So you have to move from something very rigid to something very soft, very flexible, stretchable, configurable, or even imperceptible. That means you don't actually quite feel it at all. So this is what we have come up with. If you look at this device, it's actually very simple. It has microfluidics. It is mounted on a flexible substrate, and inside it is actually a conductive liquid. The conductive liquid is made of this liquid metal called e-gain. So e-gain consists of two types of materials, iridium and gallium. Iridium is a material that we use for dental filling. Gallium is actually a trace element that we use for vitamin pills. So it has very low toxicity. So this is actually how it looks like. It's mounted on a very flexible substrate. If you look at it, there's a silver line. That itself is actually the microfluidic channel, and that is there's a liquid metal inside it. So the innovation is we are using a liquid active sensing element. Of course, you may ask, why do we use liquid metal? The reason why I use liquid metal is because it's conductive. And so we can apply a very small current, and if you were to press it and so on, it gives you a different signature. In fact, the working principle is as such. Yeah. So you have, for example, a very straight channel, and inside is the liquid metal. If you press it, you apply a small current through it. If you press it, because it's flexible, right, the whole channel will deform, the liquid gets displaced, and as a result, you get a very different electrical signal. And this is how we can sense sense forces exerting on the sensor. So if you mount it on your skin and you go about doing your activities, the sensor itself gets stretched, gets bent, and it creates different signature. So from the signature, we know what kind of activity the person is performing. And of course, we do many tests to look at the robustness and so on. So this is actually just one interesting test we did, just for fun, because my student wanted to see if it can withstand the weight of a car, or at least have a car wheel roll over it to see what happens. So if you look at this movie here, basically we initially capture 3 signals here, and when we roll over it, we get a spike. And then after that, we press it again. Actually, surprisingly to us, we found that actually it still works. There's no leakage of the liquid metal and so on. So this is actually a very robust sensor that we have here. And of course, when we developed this, we started to explore all the different applications. So it involves not only artificial sensing, where we actually incorporate our sensor into soft robotics, but also putting it on the tip of a finger for us to see if we can feel roughness, to doing rehabilitation, which I'll talk about afterwards, where we put it into the sole of the shoes for the insole of the shoes for patients to measure foot pressure. to being able to monitor grasping and finger movement, in particular for rehabilitation. And also for diagnosis, for example, measuring grip strength or even measuring finger tremor for patients suffering from Parkinson's diseases. I won't go through all of them, but I'll show you several applications. So one of the first that we tried was actually for Parkinson's disease. And here we are looking at having the ability to quantitatively measure tremor. And so when we attach the sensor onto the finger, and if we simulate tremor, we can actually on the computer be able to look at the severity of the tremor. So if we stop, it will go down to baseline. But if we start to simulate tremor again, we will see this movement. So from here, we can kind of track the tremor itself. And of course, for patients, let's say undergoing medication, and we wanted to see if there's improvement and so on, this is actually one, we talked to the clinicians, and this is possibly one tool that they can use to see if there's a significant improvement made to the patients. The other one that my student come up with is quite interesting is, all of us are using cell phones, right? And we use our thumb a lot. And surprisingly, actually, there are people suffering from injuries in the thumb because of the use of the cell phone. And so we came up with this glove to help to address that. So we use phone quite a lot, and there are people suffering from such injury. And so if they suffer injury, then we came up with this device called a wearable orthotic glove to assess thumb dynamics. And the idea is that we are able to measure movement of the thumb, even measure forces. So just to show you where the sensors are, we mounted it outside. But in general, for example, when the patient moves the thumb, we can look at whether there are improvement made to the movement of the thumb, whether there's more flexibility, and whether it's healing well. So this is one interesting gadget we have come up with to measure thumb dynamics. So we call it the thumb sense. And it's developed by my PhD student Zhu Chuan and also Zhao Ming. So the third one I'm very excited about is actually with regard to diabetes. And we know that diabetic patients do suffer diabetic ulcers. And as a result of that, some of the patients actually had to go for amputation because when it becomes too serious and becomes gangrene, they have to go for amputation. So our aim is to actually try to aim towards zero amputation. And in fact, in Singapore, every year there are about 1,000 over amputations, 1,200 or 300 amputations a year, which is quite severe. And we know Singapore has, a few years ago, launched a war against diabetes. And unfortunately for us, we are actually ranked one of the highest in terms of people contracting diabetes. And in fact, today, if you are age 65 and above, 1 in 3 will have diabetes. And then if in our lifetime, we have 1 in 3 chance of getting diabetes. And also this is related to race as well, whether you are Indian, Malay or Chinese and so on. We have some predisposition to diabetes as well. But diabetes itself has a number of complications. In fact, Professor Vernon Wu is probably the best person to talk about hypertension and diabetes. But if you look at this slide here, there are a number of complications related to diabetes like blindness, kidney failure, heart attack, stroke, and amputation. And in fact, amputation itself relates to a diabetic foot problem. And one reason why is because when someone has diabetic foot ulcers, it's kind of like a chronic wound. It's very hard to heal. It gets infected very easily. And in fact, foot problem accounts for more hospital admission than other complications, long-term complications of diabetes. And this is actually what happens is just one example of what happens when someone has an ulcer. And in Singapore, we are one of the world's highest diabetes world highest diabetes related lower extremity amputation. And in fact, there are 4 amputations a day. So 1 in 5 with lower limb amputation actually will die within a year, because majority of them are actually quite elderly patients. And so if you look at globally itself, diabetes itself, in fact, is higher than most types of cancer like breast or lymphoma. So it's a very serious problem. So what happens is if an elderly has if someone has a diabetic ulcer, they normally try to seek help in a polyclinic or GP. Normally they will consult, for example, a podiatrist. And one of the best way to treat foot diabetic ulcer is to try to minimize pressure acting on the ulcer itself. And so sometimes they recommend total contact casting like this, which the take-up rate is actually very low because if you wear the whole total contact casting is actually not very comfortable. It impedes your movement and so on. So it's probably, I was told less than 5% would do it. And then there's off-the-shelf diabetic shoes, where the shoes itself have a very soft insole, again to minimize pressure acting on the ulcer, or if you already recovered from the wound itself. But the unfortunate thing is that for diabetic patients, right, they also suffer from neuropathy, meaning when they walk, some of them don't feel pain at all. For us, if we have a small stone inside our shoes or you have a cut, we can feel immediately pain, right, when we try not to step on it. But for diabetic patients, they sometimes don't feel pain at all. So sometimes when they walk, they just keep stepping on it, even without knowing that they are actually applying a lot of pressure on the ulcer. And so there are 30 to 40% recurrence, and this is what happens. Patient suffers from infection and gangrene, and then the outcome is that they have to go for amputation. So when we look at this problem, talking to our clinical collaborators, we wanted to develop an insole that will help the patients to monitor pressure acting on the foot, on the sole of the foot. So we call it a smart insole. And we embedded our microfluidic sensors into an insole like this. And these sensors can actually track the pressure acting on the foot. And wirelessly, it can transmit the data to a cell phone. So this is a movie to show how it works. So here, my student is wearing the insole with the sensors. And as he walks, we can actually track the pressure exerting on the different parts of the bottom of the foot of the person. And so immediately and wirelessly, we can actually, using a cell phone, track the different pressure acting at the foot. And so when we came up with this device, there were many interests in this. So we started a company called FlexoSense in 2016 to actually commercialize this. And our idea is to firstly enable the clinicians to be able to perform accurate measurements of the insole pressure for better ulcer management and prevention, and also allow the patients to wear this back home. So if they are going out for a walk, and as they walk, they can actually be able to monitor the pressure acting on the ulcer. If it's too high, then they can either stop to rest or change their walking pattern such that they can avoid stepping on the ulcer.
Diabetics in Singapore have one of the highest rates of lower limb amputation in the world, and repeated pressure on foot ulcers is one of the causes.
Hi, Sophie.
Hello.
One of the reasons why they are exerting pressure on their ulcers is because when they walk, they don't actually feel pain.
Using nanotechnology, these scientists are helping patients prevent excessive pressure on their ulcers. And it's a tech that's now catching the eye of medical professionals.
So we have developed this pair of diabetic shoes with the sensors that's embedded in the insole. Where they use these shoes as they walk, we can actually monitor the pressure acting on the ulcer.
I think this technology potentially revolutionizes the way that we manage diabetic patients.
This flexible nanotech could soon be improving the lives of diabetics around the world.
So this is a movie clip done by National Geographic. They were very interested in the technology we have, and so they did a movie from here. And let me show you again another movie, how this whole thing works. So basically, we have the sensors embedded in the insole as we walk. This is an app on a cell phone that can measure pressure. If the pressure is too high, then we can alert the patients that pressure is too high. You have to take a break, sit down, rest, adjust your shoes before you start walking again. But of course, the idea is to be able to not only inform the patients to take care of their ulcers, but at the same time, we can actually be able to collect the data transmitted by the sensor to the cell phone and uploaded to the cloud. So when you upload the cloud, at the end of the week, we can generate a report and send it to the doctor. And the doctor will know for this particular patient whether they has been taking care of the ulcer or not. And that's the idea, because currently, for diabetic shoes, there's no way to track whether the patient firstly is using it, and whether they're using it, whether they are still stepping or exerting pressure on the ulcer. So we're very fortunate to get many awards and recognitions. I think one of the ones that we are pretty proud of is that we were one of the 18 startup companies from around the world to be invited to Berlin to showcase in the ID TEDx Launchpad. And so this project is still ongoing. Because of COVID, we couldn't do clinical tests or clinical trial, but hopefully near the end of the year, we can do that. So we also got a pretty big funding from this Ministry of Health actually to push this further. So the last one I want to talk about is actually the 1D sensor that we've come up with. So earlier on, I showed you it's a microfluidic patch, but right now we came up with a very simple idea, which is a microtube. Again, we fill it with liquid metal and make it into a sensor. So if you look at this particular sensor here, basically it's a microtube like this. We fill it with the E-gain, the liquid metal. And by stretching it, we can actually be able to measure forces. And after I'll show you all the different applications we can do with this sensor. But just to show you how small and imperceptible it is, if you look at this, you can't really see it until you put a magnifying glass over it, because it literally has the diameter of a strand of hair. So it's actually very small, but very sensitive. And so we came up, we characterized this initially. For example, we can stretch it, and we can measure force immediately. In fact, it's so small, we can actually string it through the eye of a needle. It can be as big as 2 times the thickness of diameter of a hair, 3 times stretchable. We can tune the sensitivity, make it very sensitive or less sensitive. And it's also washable and wearable. In fact, we follow according to the ISO standards and wash it 7 times with a 5 kilogram load of clothing in a washing machine using usual washing cycle and washing powder. And after 7 cycles, actually the sensor still looks intact and still functional. So just to show you that actually if we embed in a fabric like this or a bandage, and when we stretch it, we can also still be able to measure forces. So it's actually very useful. In fact, one of the applications is for compression bandage therapy, where we apply pressure via a bandage for patients with venous ulcer. And we wanted to measure how much pressure to exert so the nurse will know how much pressure to apply to the patient. And that was one of the applications. So just to show you, it's actually stretchable, very, very sensitive. And I will just show you a few applications. One of them is actually pulse monitoring. As you know, for us, we wear, even our tracker or our watch can measure pulse. And sometimes when we talk to clinicians, they say this is actually not the device they want. They want to measure pulse at any parts of your body. How are you going to put a watch like this, for example, and then put it against your neck or your foot to measure pulse? And so let me show you what we can do. So with this very tiny one fiber sensor, what we did was we weave it into the tip of a glove like this. And first, we measure bronchial pulse monitoring. We just put our finger on the inner elbow. And if you look at this computer screen here, we were actually able to measure the pulse wave, the waveform. So this is the pulse shape. And this is actually raw data. We also put it on the wrist as well to measure radial pulse. So again, we can measure the pulse shape here. And this is quite interesting because whenever I go to China to present it, they were asking whether we can do pa mai. Use this to do pa mai. And of course, we put on neck, we can also be able to measure the carotid pulse as well. And the last one is the dorsalis pedis pulse monitoring, where we can put it near the foot. And this will be useful for clinicians who want to do a perfusion of the microvasculature to the foot itself. So with this, just one tiny sensor, we can literally stick it on the neck or the inner elbow on your foot, and we will be able to measure the pulse at these different parts of the body. Then the other one is more related to rehabilitation and sports. where we weave a single microtube sensor onto a kinesiology tape. And the idea is, for example, for rehab, for the person they are doing bending on knees, we can actually be able to track in real time the performance. For example, when we bend our knee, we can actually, on a computer, not only know how many times the person has done it, but also how fast they have done it, and whether there are improvement in terms of flexing of the knee itself. When we talk to some of the collaborators at the rehabilitation center, they were very excited because they said they don't have to be there to monitor the patient, but it's just looking at the plot itself, they know whether the patient is doing it correctly or a number of times and so on. And the other exciting one that we are developing is a smart glove. So for those of you who have watched this Minority Report movie, you can see Tom Cruise using a pair of smart gloves to move information across the screen. And so we also weave our sensors into the glove itself. So the silver lines is actually the sensors that we weave into the glove. And over here, what we're trying to do is to incorporate this stretchable sensor into the finger so that when you bend your finger, you can get a signature. So if you look at this, because the sensor itself is very thin, very fine, we can actually easily incorporate the sensor into a very soft, very thin glove, as compared to the current commercial available gloves, which are actually much thicker. It's a bit like a bike glove compared to something like this. So we call it the Infinity Glove because we have a brainstorm, and during a time, Avengers was a movie and thought that we want to put control in your hands. So we came up with this Infinity Glove. And certainly, it can be used for a number of applications. One is in terms of gaming. Second is rehab. But let's look at gaming itself. So by just bending the finger, you can see the signature is very responsive. We can actually be able to measure signals. And one reason why we go into gaming is so partly because products like gaming products don't have to go through regulatory approval. You can get it out very quickly. Of course, we still can use it for tele-rehabilitation to enable accurate motion tracking, especially for patients who need to do rehab on the upper limb. Now, of course, if you talk about gaming itself, because we have 5 fingers, right? Effectively, one finger can perform one function. So you could either do pause, crouch, jump, shoot, or aim if it's gaming. But at the same time, through moving our fingers, we can also do advanced hand gesture like opening the map or restart or help and so on. So it depends on how we want to program each finger movement to perform a certain function. In fact, 2 fingers together can also be another function. So there are a number of different permutations that we can give to this glove itself. So let me just show you a video. It was actually launched Thursday. In fact, on Thursday we did a press briefing. So it came out in the Straits Times Online, Channel News Asia, and also Channel 8 News. So we have actually set up a company called Microchip Technologies actually to commercialize this sensor itself. Because as I've shown you, the sensors can actually be used for quite a number of different applications. And we were quite fortunate actually to win quite a number of awards as well over the last 2 years with regard to this particular sensor. And of course, hopefully we can be able to push this out near the end of the year or next year. We got some of the products we're working on. So moving forward, what would the sensors of the future be? In fact, when we were talking to a number of users, some of them are quite elderly and they may not be technology savvy. They actually will not quite like to keep wearing sensors. Maybe you can wear it for the first few weeks or months, but after that they just put it aside. So thinking ahead, I think the sensors of the future will actually be embedded into the devices that we're using every day. So be it on the bed itself, where we can monitor our sleeping patterns. Right now some of us are wearing again a watch to monitor our sleeping habits, but perhaps by embedding the sensors into the bed itself, and over time using AI, we know what the usual sleeping habits is for the person, but if it's becoming out of the ordinary, then there may be an indication of certain problems that the person may be suffering from. The other one is actually a mirror that can, we look in the mirror every morning, brushing our teeth and so on, and we can have a very special camera that can take the face and be able to track our health conditions, for example, from the tone of our skin and so on. And even toothbrush itself. If you embed sensors in the toothbrush when we brush our teeth, you will also be able to sample saliva and so on. And again, we can go for certain biomarkers that may be indication of certain diseases we may be suffering from. And of course, actually, I took this article in 2001. That's like 19 years ago. And already people are talking about this. But if you look at Glass itself, Google Glass came out with this actually not too long ago. During the time, the idea was that many of us, even myself, sometimes I forgot the name of the person, or my classmate and so on. But maybe when I wear the glass, I look at the person, immediately the name flashed across my glass. Then I won't be embarrassed not to know the name of the person. But of course, we are looking at many other applications now. And, glass actually is quite controversial because it can impede either your vision or distract you while you're walking. But people are using glasses for other applications in a more commercial setting. But one interesting project actually we were thinking about, but recently there was also a publication about this, is actually a toilet bowl. And so there's a recent publication about this toilet bowl developed by a group in Stanford. And actually there are 3 cameras in there. And of course, you think about putting cameras in a toilet bowl is a bit, very intrusive, right? But interestingly, the 3 cameras are not the cameras you are talking about. And they measure 3 things. One is when you sit on it, you measure your anus, the hole. And the researchers say is that all of us have different patterns, different shapes and so on. So if a person is sitting there, they know exactly who the person is, which is quite interesting. Second is the second camera is that you measure the stream of, when we pass urine, they can measure the stream of urine. In fact, recently there was a clinician who was asking me, can we measure the stream of urine, how fast, how much the person is passing out as an indication for some of the diseases the person may have. And the third is the camera measures a stool. They look at the combination of the stool, the texture and so on, again using that to track certain condition of the person. But I think moving forward, and which is something we are very interested in our lab, is we are putting biomarkers in it. Right now we are just taking pictures and looking at it, looking at pictures and so on. But if we can sense in the stool or urine certain biomarkers, then we can be able to detect more precisely or more accurately the condition of the person. In fact, one project that we're going to start off with is developing something like a sensor that, like a dipstick sensor into urine. But this sensor has multiple biomarkers that can track all biomarkers at one go. And so let's say if we have sensors embedded in the toilet bowl and we pass urine or stool, we can literally sample the biological data from all of us. And again, the idea is to track over time. And each of us are, as I mentioned, are individual. My data could be very different from your data. And I have my own baseline, which is also different from your baseline. And if there's a deviation, again, that might be an indication of certain condition we may be suffering from. And so this is something actually we are very keen to look into. And the reason why, again, I tell you this is because not all of us like to wear things, but if you're embedded into devices like this that we use every day, then that will be something I think we will be more keen to use. So before I end this part, I just want to acknowledge my postdocs and students who actually work on this project. They are all very, very smart, very enthusiastic and very hardworking students and postdocs. And what you have seen is actually are the different devices they have come up with. And of course, we're very grateful to our collaborators from NUH, SGH, and also even Temasek working on some of these devices. And also NUS itself, Enterprise, NUS Grid, and so on, because we are commercializing, so they are very helpful in helping us commercialize our technology. Of course, the two companies that I set up is Fluxosense and Microtube Technologies. We are continuing developing technologies in our lab, but we will actually spin out or license to these companies to further commercialize this. And also NRF and HIFES are the ones who provide the money. So the last part I want to share is actually the COVID-19 point-of-care test that we have developed in our lab. And the organizers wanted me to share something like this because these are the research that is actually funded by NUS itself and not from funding agencies. And I'm sure the money that you donated to NUS, I'm sure some of this money has been used to fund some of the research involving COVID-19 because when I received the funding, and during that time was in March, it was done very quickly. And we know we are in a big, not only national, but international or global crisis. And NUS wanted to see if some of us can help contribute towards this crisis through developing some of these test kits or other ways that can help to mitigate this crisis. So in March, there was a funding call. That's just for $100,000. I mean, normally projects like this will ask for a few $100,000 to a few $1 million. But given what we have, and given the short time that we need to develop, I wanted to see what are some of the problems that we can help to overcome. And there was a one particular challenge. In fact, now it's still the challenge, and that is that current test takes time because if you do a swab, whether in a dormitory or in a workplace, the sample will still have to be collected and sent to a centralized lab to be tested, and then the results will be made known to the person. And the whole process could take, fastest is a few hours, otherwise it will take 2-3 days. And I have also been talking to people in the US who are very interested in our test kit, and they tell me that they could take as long as 2 weeks to have the results made known to them. And that is certainly not acceptable. And the reason why we want to actually have the results known on site as quickly as possible, because so that we know what to do with the person if the person is infected. And as we open up the economy, we also want to be able to take a plane, travel, and even workplaces. In fact, one of the companies, in fact, Amazon also contacted us because they wanted to actually have some test kit that can do swabs of their employees on a very, very regular basis. You're not talking about like 1-2 weeks, maybe every 2 days or something like this. So during a time, we took this challenge and we said, okay, we wanted to develop a point-of-care test that are rapid from an hour to a few minutes, ease of use, and have a direct readout, and low cost, portable, and can be deployed on-site in various places such as airports, workplaces, and schools and clinics. And so currently, if you look at what's happening out there, there are 3 types of tests. So PCR, polymerase chain reaction test, is the gold standard. That is the current test that we are doing for the patients. It's the most accurate, but the disadvantage is take a longer time to process the sample. Second is the antigen test. It's very rapid and inexpensive, but the cons is that it has some accuracy problem. And actually, the reason is also because of the technology that's available right now. And the third is the antibody test. which looks for any antibodies that's been expressed in the person, but antibody test is kind of knowing that the person has been previously infected. It won't be able to serve as a diagnosis, as a COVID-19 diagnosis, but more to look at whether the person was previously been infected or, also the con is that there's a lot of false positive, false negative, partly also because of the technology itself that's been used. So in our lab, we try to develop two tests. One is the PCR test and one is the antigen test. So the first I want to show you is the microfluidics-based PCR diagnostic test. We call it EPIDEX, which is taken from two words, epidemic diagnostics. I thought it's a nice word to use, EPIDEX too, because we are not talking about COVID-19, because this test kit, once it's commercialized, we can use it for any other diseases, such as Ebola and so on, even tuberculosis and other respiratory diseases. So we started, actually this project started day one of circuit breaker. So in a way, we were also quite thankful to be able to come back to the lab every day to work on this device, but at the same time, we faced a lot of challenges. Firstly, supply chain disruption. Secondly, shortages of consumables, such as PCR reagents. And thirdly, all the labs are closed except maybe ours and a few other labs. So if I want to get help, it's actually very difficult to get help. I can't get someone from a closed lab to actually come back to open up their lab to provide us with the equipment. And so whatever we can, we manage, and thankfully I have a team that's very resourceful. In fact, it's a very interdisciplinary team of people who are very good at molecular biology, PCR, electrical engineering, mechanical engineering, optical imaging. And so components that we don't have, we actually look at old equipment. We actually open up the equipment and salvage some of the components to use for our system. Anything that is not, suppliers are not able to supply us or not able to quickly send it to us, we use 3D printing. For example, these are all 3D printed boxes and components. And of course, we also use our other machines to make our own chips as well. But the core component of this microfluidic device is actually this part here. So it's a toaster size. And inside there is two components. One is the PCR unit and one is the detection unit. This big box is actually just a thermal controller. Anyway, we can shrink it to very, very small because it's just a PCB inside it that we need to use. And this is just to show you the heating element itself. And the core technology is actually this microfluidic chip here. And let me just show you this movie here. Basically, the way we do is we pipette the sample into a microfluidic chip like this in the channel. And so if you look at this chip here, it can process 4 samples in one go. And so and we can do it, there are 2 methods. One is called RT-LAMP, which we can perform within 30 minutes to detect if the person has been infected. Actual PCR, we can perform up to 55 minutes. So we can actually do it within 1 hour. And normally when we take the sample, we have the virus inside it, right? We have to do what I call RNA extraction. You have to break the virus to release the RNA. But fortunately, when we were developing this, there was another medtech company that heard what we are doing and got very interested. And so we worked with them, and they have a reagent that can actually do the RNA extraction. So it's kind of a one-step process where you do a swab, you put it into a UTM, and then after that, the whole sample can then be pipetted into this device here. So you may ask what is so different about this device from other devices out there. So right now, there are close to 130 technologies that's been approved by the FDA for emergency use for nucleic acid tests. Most of them can only be performed in a centralized lab because they're all from very big companies like Abbott and so on, Thermo Fisher and so on. So they produce the machine for very big use, but there are only 4 systems that are point of care. And that's why there's a dire need for this point of care system. So there are only 4. 2 are more famous. One is called Abbott ID now, and then the other one is sulfate Expert Express, but they can only perform one sample per cartridge. So one of the advantage of our system is we can provide 4 samples in one chip. But at the same time, during PCR, sometimes we have what we call control, a positive control, negative control, and internal control, just to make sure that when you process a sample, it's performing correctly. So our chip can also be able to do that, where we have channels for control test and 1 channel for the sample test. So right now, our idea is that when we have a chip like this, first we take a swab from the patient, we put into a reagent, then we can pipette the sample into the microfluidic chip. But this has to be done in a portable biosafety cabinet so that the person who is doing it will not get contaminated or infected. So if you look at this device, actually when you pipette the sample into the microfluidic chip, it's already contained within the chip. So the sample will not be able to leak out or whatever. And to further contain it, we have a secondary containment, which is a cartridge. So the whole chip is mounted into this cartridge. And once we pipette the sample in, we can use a disinfectant to wipe the whole cartridge. And then we'll put it into the PCR module here, where we can do the PCR test. So one approach is, if you want to do multiple tests, then we can have multiple units of the PCR modules. We can do tests all in one go. Then after the PCR, we can take it out and put it into this detection unit to see if the person has been infected. So this is one approach where we can do multiple tests and be able to not only detect whether it's infected, but through a program, we can actually also be able to capture the data and then transmit it to the cloud. So it's going to be rapid, about 30 minutes to 1 hour, easy to use. Again, we can simultaneously perform many tests. It's also portable and deployable on site. The last one I will talk about is AppSense. I derived the name AB Sense from, initially we were working on a similar technology. It was supposed to be antibody sense. But if you look at this for COVID, it's actually antigen-based sensing device. And it's an antigen-based diagnostic test. So during the time in April, there's this lady called Dr. Deborah Birx, and she is the White House Coronavirus Task Force. And she said this: we have to have a breakthrough and innovation in testing. And the reason why she said this is because currently the test being done is PCR. And PCR, again, as I said, takes a longer time. It cannot really scale up in terms of mass testing. And she said that we have to be able to detect antigens and to really move into antigen tests. So what is actually antigen test? So if you look at a virus like this, on the surface, you can see there are spikes. And this is why it has the name called coronavirus, because it looks like a crown. And these are what we call S protein, spike protein. Inside the virus, there's this nucleocapsid protein called M protein. So the idea of an antigen test is to test for antigen, components of the virus, rather than for the RNA. But one of the disadvantages of antigen tests is that because we are not doing amplification, we are not amplifying the viral particles, the test has to be very sensitive. Because in PCR, you take the RNA out, you actually amplify it millions of times or hundreds of millions of times. And that's how we get the signature. So for our case, we want to detect and target the spike protein and see if we can get, be able to use this as a diagnosis. So previously, we already have a similar technology, but we use this, what we call aptamer, as an electrochemical sensor to target S protein in swab samples. And the idea is that the sensor is like a dipstick, and there's electrode. And on the electrode, we pattern these aptamers that can specifically target S protein. And the idea is we take swab sample from patient, and because we are targeting S protein, we don't have to break the virus out, because currently, there are only 2 antigen tests approved by FDA. Only 2 out of like 130 tests that's been developed. And these 2 tests targets the N protein. And N protein basically is inside the virus. So we wanted to target the S protein. So we actually have a sensor like this, put aptamers. And once we take the swab sample, we can immediately deposit the sample onto the sensor or dip the whole sensor into the sample. And then within 15 minutes, we'll get a signal readout. And so this is how we do it. Just putting the sample on it, and then within 15 minutes, wirelessly, we can actually read out on the cell phone, tablet, or PC. So we have an atomer that can specifically target S protein. We can directly test on swab sample without further processing. It's a quick readout, 15 minutes. And again, because it's very portable, it can be deployed on site. And so the idea is be able to collect sample from patients and then be able to do multiple tests at one go, and then be able to collect the data. So this test is still ongoing, but our latest result shows that actually we can now target down to what we call 10 picograms per milliliter. And so that is that's the target actually we have set ourselves. So we are actually able to reach that. But I'm asking my team to do further tests, do repeated tests to make sure that we can indeed repeatedly be able to detect down to 10 picogram per milliliter. In fact, that's what I'm very concerned with. If we can get repeated results, then I'm not too concerned that at higher concentration we won't be able to detect them. In fact, we did a range of concentrations. The higher concentrations of the viral protein, we were able to detect them now. So this is something that we are currently looking into. And hopefully in the next 4 weeks, because we started this project only after circuit breaker period, so within 2 months we came up with this. And so right now we are doing more tests, and then after this, hopefully within a month, after we get the results, we can start doing clinical tests. And of course, we are also looking into commercializing this, because this kit itself, if you can patent atomers to target other biomarkers, you can use it for other diseases as well, not necessarily COVID-19. For the Epidex, actually, it's now being licensed. So I think they are talking to a company now to license it. So the Epidex, I think right now they are looking into hopefully be able to get it out within the next few months, the next 2-3 months. So this is a team that's working on this project. As I mentioned, we have a very interdisciplinary team of people. And they really are they not only have to come back every day during the circuit breaker while other people are staying at home, but they also have to be very resourceful to get all the necessary consumables, equipment, and other resources needed for this project. But I think they are all very happy and also very proud to be involved in this project. I think being able to come up with a system like this within 2 months is actually quite unprecedented. I would say that in my whole career as a researcher, I have not done something like this before. Normally a project like this would take about a year or even 2 years to complete, but to do it within 2 months is actually very, very fast. And of course, we have to also make sure it works right, because ultimately we are not going to use it, someone else out there is going to use it. And so we have also getting other people to test the system and to convince them that the system is actually working. And of course, we're very thankful to actually NUS for providing the funding support. And as I mentioned, actually this project was funded by NUS itself, because we want to get it off the ground quickly. And initially we were given $100,000, but I quickly spent $50,000 within 1 to 2 months. And the reason is because during that time, reagents are getting more and more expensive. The cost is escalating to a point where I asked my students, I think we better cut down on the use of reagents, meaning instead of using 20 microliters, we only use 10 microliters. And the good news is that when you use small amount of reagents, we were still able to detect down to 10 copies of RNA per microliter, which is the limit. And so we were quite excited that even using smaller amount of reagents, we're still able to detect the RNA. And in fact, when we're talking to some of the companies, they are also very excited because of the this shortages in reagents and so on. You can use small amount and going to cost less per test, then that will be actually very good. So this is where we are at right now. And hopefully, next few months we can get the test out to be able to use it eventually. We got a lot of queries as well from other countries, for example, like US as well. So they are also quite excited and see if some of these technologies we develop can be deployed over there. So with that, I come to the end of my talk, and we are still in COVID-19, so please continue to stay safe and healthy. And with that, thank you so much for coming to attend this talk, and I'll be happy to answer any questions that you may have. Thank you so much. Thank you.
Thank you, Prof. Lim. So we will now have 15 minutes of question and answer. For guests who came in later, you can scan the QR code on the table. This will lead you to Pigeonhole. You can key in the password and then followed by your questions.
Prof?
Oh, yeah.
I wanted to ask, between the PCR-based test and the antigen test, which do you think has the probability of commercializing first? Because clearly the breakthrough would be very, very good for industry. For example, airline industry, for example, which have very quick tests for people to board on.
Yeah.
Just to give us an idea in terms of the rate of commercialization of these testings.
Yeah, so PCR, the Epidex system, we are already in talks with a company, almost there in terms of licensing. For the AppSense, we are still testing in our lab, to look at the sensitivity, the limits of detection, and so on. But if I look at both systems, right, so Epidex is very close to commercialization. AppSense may take a bit of time, but in terms of bringing out into a product, it's much faster for AppSense. Because it's just a matter of being able to produce the dipstick sensor itself, but the electrochemical sensor app and all this, we already have them. And commercially, they can be produced very quickly. Yeah. In fact, in the research that we conducted, anything that's available, we are not going to reinvent the wheel. We just use existing components because those are components we can get easily from the market. Anything that's more innovative and different, which is the sensor and the chip itself, those are the ones that we eventually build, develop it in the lab. Yeah.
So between the
Epidex.
and Epidex, if somebody were to choose, it would suggest that Epidex is the one which is the dipstick.
So right now I know because as I said, AppSense is still being produced, being tested in the lab. It will take a while. Yeah, but because Epidex also, because you're building a whole PCR system, although it's a small size system, that itself, if you look at in terms of engineering, you have to go through a lot of verification tests and so on. So that still will take a few months. Yeah.
What about cost-wise?
Yeah. Correct, yeah, that's a good question. So, Epidex, the PCR system, the chip is actually, the microfluidic chip is actually very low cost. Right now, we are doing laser cutting in our lab to produce the chip. But eventually, if you want to produce hundreds of thousands or millions, then you're talking about injection molding. So that will be about a few cents per piece. And the cost will be mainly in the reagent used for the Epidex system. AppSense itself, right now, if you talk about material cost, is about $10 to $20. Because we are a lab, right, and we are just buying small quantities and so on. So I would think that it should go down to below $10 if you scale it up eventually.
It's $10 versus cents.
Oh, but then because you need to use a reagent. For the PCR, the reagent would be the more costly component. Yeah. Yes.
Prof, thank you. Is there in the market a wearable today that could detect the onset of a stroke or a heart attack?
Stroke or heart attack. I know, for example, Apple has a watch that measures your ECG and so on. So. Not that I'm aware of, you talk about consumable product. Apple Watch is probably the closest right now to it. But then Apple Watch, of course, has a clause that says that it's actually not meant to tell you that there's an impending heart attack. It's just to show you maybe possible irregularities. And you still have to seek medical advice or help. So, for consumable device, I don't know of any other products that can actually do that at the moment. Yeah.
I have a question for you. One of the biggest barriers to accurate recording of blood pressure during a 24-hour period is the fact that all the current technologies available now as wearables are still based on the Space Labs device.
Space Lab.
70s.
Oh, right.
So effectively, you occlude the brachial artery.
To press it, right? Yeah, correct. Yeah, which is the cuff type. Yeah.
So it's still very basic technology from the 19th century. So how come people have not been able to invent a wearable device, maybe not necessarily brachial artery, but say a fixed artery, like a temporal artery.
That's right. Yeah.
And record 24-hour blood pressure pattern. This would be very important for the diagnosis of hypertension.
Yeah.
And also the effective treatment of high blood pressure.
Yeah.
How come no one has moved forward in this area? I don't understand.
Yeah.
The US has enormous resources and yet they can't overcome this practical problem. You get someone to wear a cuff which blows up 4 times an hour during the daytime and once per hour at night. Very disturbing.
Correct. Yeah.
Changed, and the patient naturally responds with a higher blood pressure.
Yeah. Actually, I can relate to that because I had a minor surgery before. I had to stay in the hospital for a couple of nights, and every 3 hours they'll kind of disturb me. In the middle of the night, they'll wake. They have to do this blood pressure and measuring the pulse. And if there's something, like you said, that can stick to your skin that you don't really feel it, but be able to either continuously measure it or as and when you need the readings, you can check it. That would be the best. I know that research-wise, there are many groups that are working on it. They come up with all kinds of materials and devices to be able to measure that. One of the challenges is that, as I showed you, right, we have this microtube that can measure the pulse shape. So pulse shape, you can deduce heart rate and so on. But to work out what the blood pressure is, that we still need to do careful analysis and a way to calculate. But that is actually being done right now. I know of some good friends I have who's, for example, in the US or Northwestern University. They are trying to come up with the algorithm to convert the pulse shape also into blood pressure. And if we can do that and do it accurately, then I think we are there. The only problem is many of us researchers, not all of us are keen to commercialize our technology. We publish a paper and that's it, because our KPI is in terms of number of papers published, not so much in the products that we produce, although I think universities are looking at now quite differently. So, and then we have to rely on outside companies to license it if we're not going to spin off companies ourselves. And that's why you don't see many products out there. But there are many very interesting technology that is being researched into or published out there in the literature that talks about measuring blood pressure, pulse, heart rate, blood pressure, and be able to measure pulse shape. Yeah.
So the Japanese volunteers have used oscillometry, to measure the oscillation.
Oscillation, yeah. Correct. Yeah. Yeah. That's right, yeah. And because, yourself, you're a hypertension expert. So for us as a technologist, we have to actually work with you very closely to know what are the signs or signals to look out for. And, yeah.
I'm just puzzled why it's taken so long to move away from use of the piezoelectric effect conversion of the mechanical information to electrical signal. The Japanese have exploited this.
Yeah.
So millions of machines around the world, right? Measure this, what they call pulse wave.
Pulse wave, yeah.
We still haven't moved away from this idea of occluding the arteries.
Correct, yeah.
How come you need to occlude the arteries? How come you still need to have the entire pulse wave form? How come cannot impute it from
correct. Yeah.
small oscillations?
Yeah, that's why this is something we are looking at now to be able to infer from the signals we get. I think the other thing is also from my own experience, starting a company and getting regulatory approval. It takes minimum 5, if not 10 to 15 years, eventually to get the product out into the hospital. So all the technology you see in the hospital are as a result of research done maybe 20 years ago or even 30 years ago. Yeah. It takes a long time. It's a long runway for us to see research from the lab become a product used in the hospital. Yeah.
Prof, I have a question here.
Yeah.
He says, while there is frequent application and high adoption of wearable technology in the fitness and lifestyle market, getting patients to embrace this new paradigm in the healthcare space, where devices may dictate the cost of treatment for disease management, might prove challenging. So how will your team overcome this?
So it's more in terms of the cost itself. Yeah, I mean for us, as you can see, when we develop wearable devices, there are a few considerations. Ease of use is one. Then second, of course, is also the cost. And certainly what kind of information you want to get is the third. And I mean, as a researcher, this is, there's a few considerations, but eventually when it becomes a commercial product, the company who license it or eventually commercialize it are also looking into making profits and so on. I think also part of the reason because developing technology all the way to a regulated device costs money as well. So it's not surprising that eventually the device will come out with is going to be quite costly, partly because a lot of money has been pumped into doing research and doing the regulatory approval tests and so on. And of course, they also want to make some profit out of it, and so it becomes quite costly. On our side, I mean, as I said, I have my own companies, but sometimes I want my test to only cost, let's say, $50 or $100. But by the time you reach a hospital, the hospital also added cost to the device, so eventually it may cost $200 or $300 instead of the $50 that cost us that much to make. Yeah, so it's a lot of factors coming in. Yeah.
So looking at your presentation, a lot of the emphasis appears to be on materials or hardware. Perhaps you sort of talk about the software side of it. Is it a big portion of the whole IP in your whole product?
Say again, can you repeat?
Software side.
Oh, software side, yeah.
Is it a large portion of also your IP, apart from the materials and the hardware?
Oh, yeah. So yeah, I mean, it's a good question. So it depends on the software itself, what you want to do, the software, what is the purpose of software. For example, if it's just to be able to collect data and plot it out, we already have commercially available software that we can use. But if I want to develop my own software or program to analyze the data and to sift out actionable data that could better advise the patient, then those are more proprietary software that we probably won't share with other people. Anyway, it is also very specific to the sensor that we are using. Yeah. So there are a few types of software. I mean, if it's not the generic one just to plot or just to show signals, then software itself will constitute a big part of it if it's intending to use it for our sensors to analyze the data and to provide feedback to the user. Yeah.
I have a general question here. It says, how does the wearables developed by the NUS team differentiate itself from those widely available in the market?
Yeah, so certainly if you look at the device that I show, right, it's firstly we use microfluidics and liquids as using liquid sensor. If you look at the market, actually most of them, if not all, are using solid sensors. So there's a difference there. And also that also is something that we try to protect where we file a patent for it. Also our sensor itself, we can customize it because the core technology is actually the sensor itself, but whether we want to use it for example, for the glove or for the shoes and so on, is how we eventually incorporate the sensors into this different products itself is also quite different from that available in the market. It's just that we using our sensors, we can certainly customize it. We can actually make it quite personalized to individuals, depending on what they want to use it for, or what kind of data, or what kind of signal they want to sense from the human body. Yeah.
So on the patent that you were talking, how long is it, I mean, is the patent, is it depending on the country that you want to target?
Correct.
And do you patent as much as from invention to the design?
Yeah.
So that when you commercialize,
So yeah.
Who will control the company that buys the design will control the patent?
Yeah, so initially when we invented something, we will file a patent, and the patent has to accurately describe what our innovation is. For a start, sometimes you want to cover everything, but then it will go through a patent examination, and then they'll cut your claims or they'll reduce your claims. But eventually, once you have the patent, it takes actually a few years eventually for it to be granted, if it's granted. But once you file a patent, then you kind of protect it already. And then, so in NUS itself, the patent belongs to NUS. I am the inventor, but NUS actually owns the IP, the patent. Yeah. So if I set up a company, my company still has to talk to NUS to license the technology.
So it's just a license. It can't buy it.
They can. Company can still buy, but they have to negotiate with the NUS how much it's going to cost to buy it. Yeah. But as an inventor, I will have access. I will have access to royalties. Yeah. If it's sold and the university gets some money back, then I will have access to some of the royalties as a result of the sales of the device.
How long is the patent life?
20, I think it's 20 years, about 20 years. In general, I think it's about 20 over years, 20 years around that, yeah. I know that, for example, RT lamp is already reaching 20 years, so it's almost finishing. So let's say, for example, the PCR, right? I was mentioning it can also do RT lamp. Once the patent ended, which is probably in the next few months or next 1 year, anybody can use it. Yeah. Yeah. Yeah.
Can you share your views on the vaccine?
On what?
Like the vaccine.
The vaccine,
Because it looks like it is now, on the one hand, a political battle, on the other hand, a commercial battle.
Right.
And health seems to be a little bit last in the party.
Right, yeah. Okay, I'm not really involved or expert in vaccine, yeah. But I do know that vaccine involves quite some time. So right now, I know all of us are trying, many parties are trying to push it out as quickly as possible. He has to go through all the clinical trials, right? Phase 1, phase 2, phase 3. And according to some of my, I know of people who are working on vaccines not for COVID-19, but for other diseases. And sometimes they can fail at phase 2 or even phase 3. Right now, everything seems very rosy and so on, let's say at phase 1, but it can actually fail at phase 3 after billions of dollars has been put into it. And right now, we are moving at a very fast, rapid pace in terms of vaccine development. So I'm hoping that, I think you heard news, right? Russia is probably one of the first countries now to push it out. We have to see how this phase 3 trial is going to end up. Yeah.
On the microfiber technology, are you close to commercializing? And if you were to commercialize it, maybe outsourcing the manufacturing instead of doing it in Singapore?
Yeah, I think the faster is probably the glove that I show you. That is the one that we are very close to commercialization. And manufacturing-wise, we have to outsource it. Yeah, because I don't think that we can produce it on our own if you want to scale up production. But we have the technology of how to scale up the production, to mass manufacture it. Yeah.
Do we have any other questions? I want myself. Probably share a lot of interesting applications and projects that you're working on. If any of us here is interested to take part, if there's a chance, in any of your project or to find out more, is there any way we could be involved?
It depends on in what way you want to take part in. But you're welcome to visit my lab, and come and take a look and see what we are doing and what we're working on. And certainly, as I mentioned, when our new building is up early next year, the engineering medicine building, there will be also another opportunity for you all to come, not only to look at my own research, but also research done by my other colleagues in the building. Yeah.
If not, thank you very much, Professor Lim, for your sharing, for
Thank you.
for sharing. Can we give another round of applause?
Thank you so much. Thank you. Thank you.
So, ladies and gentlemen, with that, we have come to the end of this classroom experience. We hope you have found the session insightful and enjoyable. Once again, thank you for your active participation and for spending your time with us this morning. Have a blessed weekend ahead. Thank you.